Right, good afternoon everyone. My name's Jelley. I'm the technical lead at ESR in the virology department, and I'm also a PhD candidate at the University of Otago. So at the moment I'm working on a SHIVERS-I study. And SHIVERS-I is, what we're doing is in 2020 with COVID-19 being identified and going around the globe, New Zealand moved from a suppression strategy based on our influenza preparedness plan, moved to a elimination strategy. And what this meant was we closed our borders. And what we saw was respiratory viruses decreased. And then, so we had this unique opportunity then to look at viruses being introduced into New Zealand again when the borders opened, and to have a look at their circulation, to see how they were introduced, their circulation, their drivers, what their impact, what drives the circulation, and if there was any genetic determinants on their transmissibility and their virulence. And also what we can do is have a look to see what the undiagnosed ARIs are as well. So these are the objectives of SHIVERS-I. And we identified 3 viruses to focus on first. So SARS-CoV-2, because this is what's currently circulating and causing disease in New Zealand. RSV, this is causes severe disease in children and in adults. And also, of course, influenza. So influenza, we have a seasonal vaccine, it evades us all the time. You know, at best we're about 50% VE, apart from last year, 2021. So these are the 3 viruses that we are going to focus on first. We've got different steps that we're doing, research steps. So, and we're at different stages for all 3 viruses at the moment. So we looked back to see what data sets we could have with the positive samples. So with RSV, we went back to 2016 and going through to 2022. With SARS-CoV-2, we're looking at 2020 onwards. And for influenza, we've gone back to 2012. And so this is giving us 10 years worth of data. So what we can have a look at is to see pre-pandemic, mid-pandemic. I don't know what to call it. It's not post-pandemic. So what we're going to do is a whole genome sequencing on all of them and have a look to see what we can see. So I'll start with RSV. We're further along with RSV just because it was a smaller data set to begin with, and we can, SARS-CoV-2, we had 3 sequencing facilities sequencing this virus. And for influenza, we're still sequencing. So we've got majority of the sequences for RSV. So RSV, we've got 2. We've got RSV-A and we've got RSV-B that circulate concurrently. So we've got about 1,300 RSV samples from 2016 to 2021 that we're doing whole genome sequencing on. And for 2022, we identified another 350. And so by the end of February, we would have sequenced all of these. So a little bit about why RSV is interesting is that in 2021, we had a quarantine-free travel bubble with Australia, and this caused a short, sharp outbreak in New Zealand. So in orange, you can see 2019. This is a normal circulation for RSV. 2020 we had nothing, but in 2021 we had a big outbreak. Oops. And just to have a look to see what was circulating and who was affected. So RSV-A was the majority, and like normal with RSV, it was the under 2s that were mainly infected. So what we've been doing is we've sequenced all of 2016 to 2021. So I've been working with Jordan Douglas at the University of Auckland, and we've just been looking at different ways to analyse all of this data. So we've worked with a subset of data. which is what you're seeing here. It is an estimate at best, but using the same methodology as Dr. Gemma Gagan used in her paper of 2020 looking at the introductions of SARS-CoV-2, we've looked at the introductions of RSV. So this is RSV-A, and we can estimate the number of introductions per day over so many years. So what we can see is that there was no introductions in 2020, and in 2021 there were a lot of introductions per day. And we've done the same for RSV-B, which was a lot less in 2021. And we've used a Bayesian software, so Beast software, to create this phylogenetic tree. So this is calibrated by time, and what you can see at the very top is it's been introduced into New Zealand, and we've got this very big cluster of local transmission of RSV-A, and we've got some. Is this the pointer? Yeah, and so this is the very big outbreak that was caused, and we've got these little local clusters here. So we've multiple introductions into New Zealand over 2021, and just remember that this is a subset of data. So it looks a bit interesting up the top. It looks like, this strain has actually been circulating in New Zealand since 2012. But we know that this isn't true because with the Australian data, they had an off-season outbreak of RSV, and we know that they had a lot of RSV in about December to April last year, and this was introduced into New Zealand in April. So for RSV-B, there was less introductions into New Zealand, and they clustered together, about 2 or 3 clusters right up the top there. So the next steps with RSV is we will actually want to really have a look at this analysis of all these genomes. So we want to add the 2022 genomes into it. We want to rerun the Bayesian analysis with a lot more global data so we can really see those introductions and where they came from. We want to do a deep dive analysis, so we want to integrate all the metadata that we have and to put this on with the genomes. So we want to have a look at outbreaks with those little local clusters that you see. Why didn't they go further? Why did the one up the top with RSV-A, why did that, why is that so big? Where was that? Who was that affecting? Looking at the demographics, were they all less than 2, those ones? Things like that. Okay, for SARS-CoV-2, we've had 2 papers published. The first one was looking at the Delta outbreak, which was August 2021. And this was a very big outbreak for New Zealand. It went from New Zealand being under the elimination strategy to a suppression strategy, just because there was no way we could stop Delta from infecting people, all those transmission. And also we had Delta variant to about February 2022 when Omicron came through, and the second paper was just looking at the different Omicron variants when they came in through New Zealand. So we've got 2 studies under SHIVERS for SARS-CoV-2. So we're looking at the household transmission study, which you heard Naira talking about. And so this was very intensive. This was swabbing every 3 days if you were positive. And we've done all of the whole genome sequencing from this, and we're just cleaning up that data. And what we want to see from here, so one person was being swabbed every 3 days, so we've got multiple genomes for the one person. We want to analyse these. We also want to analyse the transmission dynamics within that family also. Did it change in the genome? We also want to see if there was reinfection or if a household was infected with multiple variants. So during this household study, so in February we had BA1, we had BA2, and later on in 2022 we had BA5. So we've also got the acute study, and this is everyone in the SHIVERS 2, 3, 4 who was positive with their whole genome sequencing, so not the transmission study. So we don't have multiple for the for the one person unless they were reinfected. Which leads us to influenza A and B. So my husband, when I became a PhD student, he told me he's become a PhD widow. And I don't think that's very true yet, but I can see that when the influenza data is finished, that's actually going to be quite true. I can see myself sneaking away from boring family events to have a look at this data. So this is huge. So we've got 10 years worth of data here. So we've got 4,000 influenza A's and we've got 1,250 influenza B samples, and we will add more to this. So all the samples have been extracted, they've been aliquoted out, we've sent them for whole genome sequencing, and that's what's happening at the moment. So Hugh's team has done an amazing job. They've really got this optimised now, and everything will be completed about midway this year. We've done a little bit of influenza A H1N1 for 2022, and looking at the phylogenetic tree, it is closely related to Australia, which is just what we can assume most times. I think with being so close to Australia, I think we're going to infect and reinfect everyone all the time. So next steps for influenza, we're going to finish the sequencing. We're going to do a phylogenetic analysis, a bit like what we've done with RSV A and B. We will do that for all of the subtypes and the lineages for influenza. We're going to do a deep dive analysis, so integrate the metadata into with those genomes, and we're going to see what we can see. We're going to have a look at outbreaks. We're going to have a look at who's infecting who, and things like that. We've also got the household transmission study, and that's exactly like what we've got with SARS-CoV-2. Multiple genomes for the one person. We can have a look and see if the genome's changing and have a look at the dynamics within that household as well. Okay, gosh, I really talked through that really fast. I'm amazed. And just my thank you page, the font for this gets smaller every single time because we keep adding people in, and everyone, it's really an accumulation of a lot of people's hard work. And I want to take the opportunity to really thank the Well Kiwis clinical team led by Tinika Jennings and the Well Kiwis study team led by Ruth Seeds. The household transmission study is just going to be an amazing study. And it was an intensive lot of work, a lot of swabbing, a lot of sending out of swabs and bringing them back in, keeping the lab really busy. So I just wanted to highlight their hard work with that.